Hello, everyone, and thank you for coming to our June Medical Mycology Trainee Seminar Series. We have two fantastic speakers today, but before we get started, just a couple of announcements of interest that might be of interest to the community. So the next Mycotalk series is coming up at the end of the month, the fourth Thursday on June 30th. And the speakers are Myri Nover and Zin Lin. Both should be fantastic, and it's free to sign up. That's an online series, so I'll put the sign up information in the chat. Also, the Medical Mycological Society of the Americas is having their annual symposium. It is next week, June 14th, I believe. Yes. And that is also free to sign up. There will be short talks from all of the award winners for this year, as well as the new members of the American Academy of Microbiology. So that should be a great series as well. So I'll put the information in the chat. Also, we'll take questions after both speakers have presented. So please put your questions in the chat and the moderators will be moderating it. And so our first speaker this morning is Dror Assa, who earned his Bachelor's of Science in Chemistry and Biology at the Hebrew University in Jerusalem. He then completed a master's degree in biological chemistry from the same institution while researching influenza A. Dror then moved to the University of California, San Francisco for his PhD, which will very soon be defending, and he's currently on the job market looking for positions in the biotechnology and pharma industries. So, Dror.
Okay, one second, let me share. Thank you for the introduction. One moment. Okay, can you see my screen fine? Okay, so hello everyone. Thanks again for the introduction and thank you for inviting me to speak to you today about my dissertation project. It's a really great opportunity. So today I'm going to tell you about as I mentioned, my project about how Cyclic AMP triggers filamentation through a distinct transcriptional program in the human pathogen Histoplasma. So, one second, let's progress. So let's have a laser pointer. Okay. It is not moving. Okay, this is okay. So I'm going to start by giving you a little bit of background about Histoplasma and the project, and then I'm going to tell you about my data about how exogenous CM promotes filamentous growth in Histoplasma. I'm going to tell you about a little bit about the results of a transcriptional profiling experiments that I conducted. And then the follow-up experimentation that from this experiment in which I identified two genes that are necessary for this response to Cyclic AMP. So I'll start with giving you a little bit of background. So Histoplasma is what is called a thermally dimorphic fungus. What that means is that it grows in the soil as hyphae that produce spores. And when those spores are aerosolized, they can reach the alveoli of the lungs of mammals' lungs, including humans, where the elevated body temperature causes them to transition into the budding yeast form. And this causes a disease called histoplasmosis. And here I'm showing you the distribution of this disease, and you can appreciate that it's very common in North and South America, as well as in parts of Africa. But there are also outbreaks in other places in the world, and they're causing severe morbidity and mortality burden. In the lab, we're able to transition we're interested in the regulation of this transition between hyphae and yeast, and we can replicate this transition by just switching the temperature of the culture between 37 degrees Celsius to promote yeast growth, or room temperature growth conditions to promote hyphal growth. And I know that there are at least a few people in this audience that are very familiar, for instance, with the Candida transition between yeast and hyphae. I just want to emphasize at this point that this is the opposite direction. So Candida actually transitions into the filamentous form at 37 degrees and grows as yeast at the lower temperatures. So just bear that in mind. And as I mentioned, our lab has been really interested in the regulation of how this transition is regulated. And one of the main approaches that we've used in the past to learn about this transition is genetic approaches. such as a forward genetic screen that that allowed us to identify with follow-up experiments 4 transcription factors that are each required for the yeast phase. So what I'm showing you here is the results of the knockouts of each one of those genes. So each one of them is required for yeast phase, hence the name RIP, required for yeast phase. And as you can see, Histoplasma wild type grows as yeast at 37 degrees and transitions to the filamentous form at room temperature. However, depletion of each one of those genes causes constitutive filamentous growth at either temperature. However, follow-up experiments also identified that each one of them is a master regulator of the yeast transcriptional program. However, we don't really have good candidates at the moment for regulators of the filamentous form of Histoplasma. So coming into this project, I asked the question, can we use other approaches, namely chemical approaches, to find regulators of the filamentous growth form? How that would look? So the idea is that we have Histoplasma growing as yeast at 37 degrees. And when you add a chemical to this to the media, those cells would transition to the hyphal form despite the elevated temperature. And if we want to imagine kind of a general or simplistic pathway that would enable that, then this chemical would induce a gene, and this gene would somehow repress the 37 degrees program that includes the RIPS to allow for hyphal growth. And obviously, what we're really interested in here is what is this gene that allows for filaments to grow. So what could a chemical like that be? We already have some hints from old literature. So here I'm showing you a figure from a paper in 1981, in which they tracked the levels of intracellular cyclic AMP in Histoplasma growing as yeast at 37 degrees or transition to the mycelial form or hyphal form at 25 degrees. What you can appreciate that starting at 72 hours, which is roughly when the budding yeast start to grow as filaments, that the levels of cyclic AMP go up in the inside the cells. Now, cyclic AMP really is important in every living organism on this earth, and it's been it's a secondary messenger and has been studied extensively in a lot of different organisms. One of the organisms that where it's been studied really thoroughly is Candida albicans, and a series of studies have shown that adenylate cyclase really integrates a wide variety of signals that each one of them is promoting filamentation. And adenylate cyclase produces cyclic AMP, which is broken down by phosphodiesterases. But when cyclic AMP levels are high, it signals through protein kinase A to activate a transcription factor called EFG1, and that is antagonistic to a transcription factor called Dror. And those two transcription factors are really interesting, especially because to us, because their orthologs in Histoplasma, Stu1 for EFG1 and RIP1 for Dror have already been on our radar. I already mentioned RIP1. Stu1 is also relevant in other fungi for developmental decisions, such as conidiation and fermentation. So both of them have been on our radar. And this allows for the activation of EFG1 permits filamentous growth. And in fact, when you add exogenous cyclic AMP in Candida in the form of this non-degradable cell-permeable analog called dibutyryl CAMP or DBCAMP, Candida transitions into the hyphal form. So the hypothesis, by analogy to what I've just shown you, is that room temperature somehow activates adenylate cyclase, and the elevated amount of CAMP signals through protein kinase A to activate the orthologous, the Stu1, which is orthologous to EFG1, and this could be antagonistic to the RIPS and promote filamentous growth. Okay, so now I've given you all the background that you need to, and I can tell you more about what I actually did. So the first thing that I wanted to see is whether or not I can get the same results similar to Candida in Histoplasma. So I took different cyclic AMP analogs. Here I'm showing you dibutyryl CAMP as well as a different analog called 8-CPT-CAMP, and I added them to growth media of Histoplasma growing as yeast, as you can see here in the vehicle control, which is just water. And what you can appreciate here is that the addition of either analog of Cyclic AMP promoted filamentous growth within 2 days, despite the elevated temperature. You don't even, and this is not unique to those analogs, so there are other ways of affecting the levels of Cyclic AMP. So here, once again, I'm showing you Histoplasma growing as wild-type Histoplasma growing as yeast at 37 degrees. And adding Cyclic AMP alone, regular Cyclic AMP, doesn't cause a very strong effect. And that's not surprising because Cyclic AMP is broken down very readily by phosphodiesterases and is not super cell permeable. However, if I add an inhibitor of phosphodiesterases called IBMX, I get some induction of filamentous growth. And this is, and I have an additive effect if I add both IBMX and exogenous Cyclic AMP. So this again emphasizes that exogenous Cyclic AMP, either in its regular form or as analogs, can promote filamentous growth. So our lab also has identified a yeast-locked mutant, which we named SG1, which does not transition to the filamentous growth in response to room temperature. So once again, Histoplasma grows as yeast at 37 and filaments at room temperature, but the SG1 mutant continues to grow as yeast. I wanted to see if this, to test whether this mutant responds to Cyclic AMP. And what I found was that similar to its lack of response to room temperature, it also does not respond to Cyclic AMP. And this is interesting because it allows us to have kind of a tool to assay the, a tool to look at what the effects of Cyclic AMP could be on the wild type by comparing it to this non-filament mutant. So to summarize this section, Cyclic AMP and its analogs induce hypogrowth in Histoplasma at 37 degrees, and the SG1 mutant does not filament in response to Cyclic AMP. Okay. So the next thing I wanted to know is how does the transcriptional program in Histoplasma change in response to Cyclic AMP? So I took those two strains, the wild type and the SG1 strain, and I grew them either with the water vehicle or added DBCAMP. And as you can appreciate here, just as before, wild type filaments in response to DBCAMP and the SG1 strain does not. I'm not going to go into all of the analysis that we've done. I'm actually going to jump to the bottom line in this time. But the comparison between those 4 conditions that I've just shown you allows us to identify 2 interesting groups of genes. And at this point, I also want to thank Mark Voorhees, who is a bioinformatics specialist in our lab, who's been instrumental in analyzing these results. So, as I mentioned, the comparison between those four conditions allows us to identify groups of genes that are of interest. And the first group I'm showing you here is a group that we named yeast-associated genes. And we named them that way because they're downregulated. So what I'm showing you here is a heat map, right? Blue, each row is a gene, each column is one of the conditions that were done in triplicates. And what you can see here is a group of genes that are downregulated in wild type in response to DBCAMP. So as you can see, this column is very blue, but are upregulated in SG1 either with or without DBCAMP. So these columns here are yellow. So these are genes that are downregulated when it's filamentous and are upregulated when it's continuing to grow as yeast. And that's why we call them yeast associated. When we look at the genes that are in this group, we see genes that are really relevant mostly to the virulence program of Histoplasma, which is not surprising because yeast is the virulent form. For instance, every gene here that you can see that has the beginning CID relates to iron acquisition. There are catalases, CATP. CBP1 is a known virulence factor, and there are other secreted factors and genes that have to do with metal metabolism in Histoplasma. But what's not in this group is a lot of transcription factors and regulators or signaling molecules. However, if we look at the opposite group to that, which is the filament-associated genes, so again, these are genes in this case that are upregulated in wild type in response to DBCAMP and are downregulated in SG1. We get actually kind of the opposite, a very different group of genes. So we get about half a dozen transcription factors. For instance, STU1 is here, YAP1 is a transcription, and AP1 transcription factor. We get metabolic genes such as ZWF that has to do with pentose phosphate, the pentose phosphate pathway, and we have other signaling molecules such as HOG2, which is up here, which is a MAP kinase or a pseudo MAP kinase. So these genes are, we have a lot of signaling genes and metabolic genes. And this is really interesting because, as I mentioned, we're looking for regulators of the filamentous form of histoplasma. So the fact that there are a bunch of transcription factors here really makes this set of genes really interesting to us. I chose to follow up on 2 genes in this group. The first one is STU1, which I've already introduced before. And the other 2 are FBC1 and PAC2. And I'll talk to you a little bit about them. So STU1 is an abscess transcription factor. It's upregulated in room temperature, at room temperature in histoplasma. It's orthologous to Aspergillus STUA and Candida EFG1, which is required for hyphal growth in Candida downstream of CM signaling, as I've told you. FBC1 is a C2H2 transcription factor. It's orthologous to Aspergillus lib-C, which is necessary for remediation in Aspergillus nidolensis. And PAC2 is a Whopper family transcription factor, which is upregulated at RT at room temperature in histoplasma. And in Schizocytomyces pombi, it regulates sexual development, which is another interesting developmental program. So to summarize this, Cyclic AMP induces a distinct transcriptional program in histoplasma at 37 degrees, and comparing the effects of CAMP to the transcriptional program in a non-filamenting mutant, that's the SG1 mutant, allows us to identify sets of hyphae and yeast-associated genes. STU1, PAC2, and FBC1 are transcription factors that are induced in response to Cyclic AMP that are downregulated in the non-filamenting STU1. So from here, I'm going to tell you about my follow-up work on those genes. So just to remind you, the hypothesis that we had was that room temperature induces adenylate cyclase to produce cyclic AMP. This signals through STU1, which is antagonistic to the RIPS and promotes filamentous growth. And as I mentioned, STU1 has been on our radar for a while. And one of the reasons is that we know that its expression is sufficient to promote filamentous growth at 37 degrees. So here I'm showing you STU1 overexpression of STU1. You can see how filamentous it is, while the control plasmid continues to grow at 37 degrees. So next, I wanted to see whether or not it's necessary for room temperature or cyclic AMP-dependent filamentation. So I created a STU1 mutant using CRISPR-Cas9, and I grew it either at 37 degrees or room temperature, or grew it at 37 degrees and added either water or DDCM. And what you can appreciate here is that this is I didn't really see an effect, meaning STU1 behaved very similarly to the wild type. It filamented in response to room temperature as well as to DDCM. So STU1 is not actually necessary for room temperature or CM dependent filamentation. What about PAC2 and FBC1? So I did here I'm showing the experiment in which I added DDCM to those. I created those mutants as well and grew them with either water or DDCM. And as opposed to STU1, here you can see that both of those mutants do not respond to CAMP by filamenting. So each one of them individually is necessary for CAMP-induced filamentation. What about room temperature? So to my surprise, both of them filamented just fine in response to room temperature. So I'm showing you here kind of a short time course in which I grew a wild-type histoplasma as well as the PAC2 and the FBC1 mutants at 37 degrees and transitioned them to room temperature. And you can, I think you can appreciate here that all of those snapshots look very similar to each other. They're all there. These mutants had no problem filamenting in response to room temperature. So they're not individually necessary for room temperature filamentation. That doesn't mean that they don't have interesting phenotypes at room temperature. So I'm showing you here, kind of as a side note, the wild type actually produces a large number of conidial spores. Those are asexual spores. There are 2 kinds, macroconidia and microconidia. And on Saborod, histoplasma produces a large amount of especially the macroconidia. However, PAC2, FBC1, and STU1 are each unable to produce normal macromedia on subarachnoid media. So even though they are those mutants filament fine at room temperature, they have other developmental defects that would be interesting to study further. So what about sufficiency? So to test whether or not PAC2 and FBC1 expression is sufficient to induce fermentation, I created overexpression plasmids. So here I'm showing you colony pictures. Here on the left, I'm showing the gross morphology of the colony on the plate. And on the right, I'm showing a microscopy picture of kind of the edge of the colony. So the dark area here is the mass of the colony, and here's the plate. The bright area is the plate. So each one of those mutants, as I've shown you before, grows as yeast at 37 degrees, as also is seen by this gross morphology of like glossy raised colony. And you can appreciate that the edge of the colony is very smooth, which is again consistent with yeast growth. However, if I overexpress PAC2 or FBC1, I get different degrees of filamentous growth. So PAC2 overexpression, the colony itself looks very fuzzy. And if I look under a microscope, I can see all the filaments kind of protruding out of the edge of the colony. And with FBC1 overexpression, the morphology phenotype is more subtle. So the colony is not very fuzzy, it's more wrinkled. So this is kind of like an odd aberrant shape. But and when I look under a microscope, I can see that maybe there is still a smooth edge, but there are filaments that are protruding out of it, but maybe not as filamentous as PAC2 overexpression. Overexpressing PAC2 and FBC1 in their respective mutants kind of gives partial filamentation phenotypes as expected. But what's also interesting is that if I overexpress PAC2 and FBC1 in the other mutants, so here I'm showing you PAC2 overexpression in the FBC1 mutant and vice versa, FBC1 overexpression in PAC2, I get filamentous growth if I overexpress PAC2, but I don't get, I get this wrinkled morphology, but I don't get filaments protruding from the colony when I overexpress FBC1. So just to iterate on that, PAC2 is sufficient to produce filamentation at 37 degrees, but FBC1 depends on the presence of PAC2 to produce those filaments, which might indicate that PAC2 is could be downstream of FBC1. So to summarize this section, as I told you, STU1 is not necessary for CAMP or room temperature for induced filamentation, even though it is induced by cyclic AMP and room temperature. PAC2 and FBC1 are each necessary for DDCAMP-induced filamentation, and PAC2 and FBC1 are individually not necessary for room temperature-induced filamentation. An overexpression of either PAC2 or FBC1 is sufficient to override the normal yeast morphology at 37 degrees, but PAC2 is sufficient even in the absence of FBC1. So here I'm showing you kind of like a model of these results. So cyclic AMP induces the expression of these three genes, and they have to, in order to promote filamentous growth, they have to somehow repress the RIPS program. In the interest of time, I didn't go into these results, but we know that I've tested and seen that despite the filamentous growth, I don't get depletion of each one of the RIPS at 37 degrees, which would indicate that this inhibition of this pathway could happen downstream of that. And then finish very soon. So this leaves some open questions. The first one is CAMP necessary for the yeast to high flow transition? Two, the other question is, are STU1, FBC1, and PAC2 redundant for room temperature induced fermentation? Because each one of them individually is not able to, deletion of each one of them individually is not able to prevent room temperature induced fermentation. Third, are there other transcriptional regulators of high flow morphology that are necessary for fermentation? There are about half a dozen transcription factors, as I mentioned, in the high flow associated form. So that could be other players in this program. And third, and fourth, what is the regulant controlled by STU1, FBC1, and PAC2? So with that, I want to thank my lab, and especially Anita, who is a wonderful mentor, as well as Mark, who's been, as I mentioned, really helpful in analyzing all of the transcriptional profiling data, and the entire lab for their helpful suggestions and discussion, as well as some previous members that made this project possible. And with that, that's all I have for you today, and I would be happy to answer any questions.
All right, thank you very much for the great talk. Always makes me happy to see nice genetic pathways worked out. And so please enter any questions for Dror in the chat. And now we'll move on to our second speaker, Maureen Hester. And Maureen is a PhD student at the University of Massachusetts Chan Medical School. Before that, she earned her Bachelor of Science in Biology and Biotechnology from Worcester Polytechnic Institute and a Master's in Pathobiology at the University of Connecticut. And now she is a member of Stu Levitt's lab, where she's working hard to solve the problem of the lack of vaccines against fungal diseases. She anticipates finishing her PhD soon and is about to start looking for postdocs. Maureen?
All right, thank you. Are my slides all right? Are they?
They're great.
Okay, perfect. All right, so good morning. Yeah, I'm Maureen Hester, and I'm a fifth-year grad student at UMass Medical School, and I'm in the Immunology and Microbiology program. And I am doing my thesis research with Stu Levitt's and Charlie Specht. And I have two main projects that I'm working on. One is in collaboration with Jenny Lodge's lab, which is now at Duke, and that's using Cryptococcus neoformans and Gadii co-infection as a tool to investigate defects in vaccine-mediated protection against specifically Gadii R265. And that's using a live attenuated vaccine. But today I'll be focusing on my other project, and I am really excited to tell everyone about antigen cross-reactivity following recombinant protein vaccination. Oops, why won't it go forward? There we go, sorry about that. Okay, so cryptococcosis is an infection that's caused by Cryptococcus neoformans or Cryptococcus gadii. And these are soil fungi that can be found in pigeon guano and in decaying wood. They're around 5 microns in diameter, so they're small enough to easily be aerosolized and inhaled, at which point they can lodge themselves in the alveoli in the lungs. And from there, the crypto can disseminate to the central nervous system and cause cryptococcal meningitis. Cryptococcal meningitis is responsible for about 15% of AIDS-related mortalities, killing almost 200,000 people each year. And while I'm sure this crowd is pretty well versed in the importance of fungal vaccine development, I do want to point out that there are currently no licensed antifungal vaccines. And even though a recent review states that over 45 different types of cryptococcal vaccines have been created and tested in mice by various research groups, none of the experimental crypto vaccines have made it to clinical trials yet. So our lab is one of the groups working to try and develop a cryptococcal vaccine. And we have a well-established vaccination challenge model in mice using Glucan particles. So Glucan particles are both an adjuvant and an antigen-presenting cell targeted delivery system, and they're basically the hollowed out cell walls of baker's yeast. We express our proteins in E. coli and purify them before they're loaded into these GPs. And the vaccines are delivered subcutaneously every other week. 2 weeks after the third vaccination, we administer an oral tracheal challenge, which inoculates the crypto directly into the lungs. And this challenge is lethal in unvaccinated mice, with the unvaccinated mice will die after 3 to 4 weeks. And the crypto will have disseminated from the lungs to the brain, similar to human infection. We measure survival until 70 days post-infection, at which point most of the vaccinated surviving mice have cleared the fungal challenge from their lungs. So we wanted to use the Glucan particle system to screen subunit vaccines composed of recombinant cryptococcal antigens. And to start the screen, we had to establish criteria for what we thought could make the best vaccine candidates. We chose to focus specifically on extracellular proteins, whether they were cell wall or secreted, thinking this would improve the chances of immune detection because they're likely the most exposed. And so we picked our extracellular proteins based off 3 main categories. It's those known to be immunogenic, those that were abundantly transcribed during human infection, and those that were present in protective alkaline extracts. We had also defined additional favorable criteria. So for example, we wanted antigens with minimal human homology to try and decrease the risk of autoimmune reactions down the line, or those that had really abundantly transcribed Gadii orthologs, hoping to get a potential pan-fungal vaccine. And we also got a little bit speculative with it. We thought that antigens that belong to protein families might actually make promising candidates because there could be a chance for cross-reactivity between family members, which in theory could yield more immune targets and improve protection. So with those criteria in mind, we selected and we tested 23 different protein antigens as Glucan particle vaccines in both BALB/c and Black 6 mice. And we found that 12 protected either one or both mouse strains that we used. So looking at the 12 that were protective, we saw that 9 of them do share homology to other proteins within Cryptococcus. And of the 11 that don't protect, only 4 belong to protein families. So we had the majority of the non-protective antigens being the ones without homologs, but the majority of the protective antigens did have homologs. So this got us thinking that maybe there actually was something to the idea of selecting for protein families. And with that, we set out to answer the question of, can antigen cross-reactivity between family members improve vaccine-mediated protection in our mouse model of cryptococcosis? To start to answer this question, we had to first establish a model to test the potential benefit in terms of survival. So we started by using one of the protective vaccine antigens that does not have any cryptococcal homologs. MEP1 is a secreted metalloprotease, and it's the 3rd most transcribed cryptococcal gene during human infection. And like I said, it doesn't have any homologs within the neoformans proteome. So we know that when we vaccinate with GP MEP1, we get protection against wild-type cryptococcus. So the hypothesis was that if you delete the antigen and then challenge the mice with the vaccinated mice with the deletion strain, the vaccine won't work anymore because the antigen the immune system was primed to recognize isn't there. The protein doesn't have any homologs, so there shouldn't be any potential for other antigens to be recognized and maybe pick up the slack a little bit in terms of kicking off the adaptive immune response. This probably seems a little straightforward immunologically, but it's actually not that easily demonstrated in vaccine models of other pathogens. So using the vaccination and challenge model I described before, we tested the system in BALB/c mice. In an unvaccinated mice, a wild-type infection, which we use strain KN99, the mice will succumb by 31 days. We also tested the strain that lacks MEP1, which is the red dotted line here. And you can see the unvaccinated mice die within a similar window as those that were given the wild-type challenge. Vaccination with MEP1 gives us about 50% survival against the wild-type crypto, which is the bold black line. But if we delete MEP1 from the challenge strain, which is the bold red line, we lose the protection that was induced by the vaccine. And there is not a difference in survival between unvaccinated and vaccinated mice that were given the MEP1 deletion strain. So this shows that the protection we're seeing is specific to our antigen. It's not due to reactivity of like the adjuvant system. And it also demonstrates that a single antigen can yield protection against a bug that's transcribing well over 6,000 genes. So now we could really start to dig into our question. And we have all these protective antigens that are members of different protein families. So is the protection that we're seeing in part due to sequence similarity that allows for immune recognition of more targets? So to look into this, I'm using the chitin deacetylase family of protein. And the family alignment is shown here. The legend on the right color codes how many of the family members an amino acid is conserved across. The CDAs are enzymes that deacetylate chitin into chitosan, both of which are important for cell wall structure and function in Cryptococcus. And 3 of 4 of these family members are protective in our vaccine model. So CDA1, CDA2, and CDA3 are protective as vaccine antigens. But FPD1, which is sometimes called CDA4, is not protective. We have single gene deletion strains for each of these, and the single gene deletion strains still express the other protein family members. And I will point out that if you delete CDA1, 2, and 3, you get a chitosan deficient triple mutant. And this is actually a very successful live attenuated vaccine, which a lab has worked with Jenny Lodge's lab on for quite some time. So we tested survival following vaccination with either CDA1, CDA2, or CDA3. And I'll start with the CDA1 results here. So looking at the dotted lines, which are the unvaccinated mice, you can see the deletion strain for CDA1 does extend survival by a few days compared to wild type, but the mice still succumb to infection by 35 days post-infection. If we vaccinate with CDA1 and then challenge with wild type, we get 100% survival, which is what we expect with this vaccine, and that's the bold black line. But if we use the same CDA1 vaccination and challenge with a CDA1 deletion strain, we get an intermediate curve, and that's shown by the bold red line. So this curve is statistically different from the vaccinated group challenged with wild type, but it's also significantly different from the unvaccinated group challenged with the deletion strain. So vaccination in this context isn't providing the same level of protection that you see following wild type challenge, but there is a distinct benefit compared to the unvaccinated groups. And we also see this trend with Cda2 vaccination. Cda2 vaccinated mice challenged with the Cda2 deletion strain survive much better than the unvaccinated mice do, but they don't survive as well as the vaccinated mice that got the wild type challenge. And on the right, we have Cda3, which is the least protective of these three vaccines. And there's no difference between vaccinated groups here, whether they were challenged with wild type or with the Cda3 deletion strain. The protection being the same in Cda3 vaccinated mice, whether or not Cda3 is present in the challenge strain, kind of raises the question of whether it's actually reactivity to Cda1 or Cda2 that's mediating the protection that we do see. Overall, though, these data demonstrate the cross-protection by family antigens within the chitin deacetylase family of proteins. Obviously, the next thing we wanted to know is what's mediating this cross-protection. So to look at the B cell response, we did Western blots on mouse serum, and I put the recombinant CDA proteins, just like we would use in our vaccines, onto an STS page gel. And we use recombinant ova as a control here because we do express all our proteins in E. coli. So this should give us an idea of any background generated by extraneous E. coli proteins that might still be there after purification. And this STS page gel is the reference gel for all the subsequent blots I'll show. So we did Westerns to determine serum IgG for each of these proteins. And first we have unvaccinated control serum, and there's no reactivity to any of the recombinant proteins. We also used Glucan particles loaded with mouse serum albumin as another vaccine control. And again, there's no reactivity to the proteins, even using a yeast-based vaccine platform. Serum from mice vaccinated with CDA1 gives an IgG response to CDA1, but you can also see some reactivity to CDA2 here. And CDA2 vaccinated mice make IgG against CDA2 and also CDA3. CDA3 vaccination actually gives you IgG that binds each of the 4 family proteins. And we see cross-reactivity with FPD1 as well. And just know that none of the serum has any reactivity with the recombinant ova control. So we are seeing a cross-reactive antibody response within the chitin deacetylase protein family. Interferon gamma is known to be very important in protection against cryptococcus. So we wanted to look at the interferon gamma response and whether or not we saw cross-reactivity following vaccination with the CDA family proteins. So to do this, we use an assay that Mrna, who is a postdoc in our lab, developed, where we vaccinate mice and then do ex vivo stimulation of their splenocytes to measure interferon gamma production. So for this assay, we harvest the spleens 2 weeks after the final vaccination, and we culture 10 to the 6 cells per well with 10 micrograms per mil of recombinant protein. And again, here we use recombinant ova as a control. These are then incubated in tissue culture conditions for 3 days before we measure the interferon gamma in the culture supernatant. So I'll start with CDA1 here. The vaccine antigen is listed on the top of the graphs with each of the stimuli listed underneath the x-axis. In addition to unstimulated groups, we have the recombinant ova setting the level of background created by using proteins purified from E. coli. So anything listed as statistically significant means it's significantly different than the ova stimulation for that group. So when mice are vaccinated with CDA1, we get significant production of interferon gamma by splenocytes following ex vivo stimulation with CDA1 protein. And that's the trend for each of the vaccines. Splenocytes from CDA2 vaccinated mice produce interferon gamma in response to stimulation with CDA2 protein. Same with CDA3 and with FPD1. But if we go back to CDA1, we also get significant interferon gamma production in response to stimulation with CDA2. And this is also the case with CDA3 vaccination. So splenocytes from Cda1 or Cda3 vaccinated mice are also producing interferon gamma after being stimulated with Cda2 protein. So far, survival studies have shown cross-protection within the CDA family, and we're seeing a cross-reactive interferon gamma and IgG response that could be contributing to this. But there's another thing to consider in generating cross-protection, and that's the expression of vaccine antigens in vivo. Sequence conservation throughout the family is obviously important for the ability to cross-react, but we also need there to be enough of the antigen present during infection to make those interactions possible. So this is a graph that I put together using cryptococcal transcript data that had been uploaded to GEO, and the info for the data sets and the associated publications are in the bottom right corner. And I really appreciate these groups making their data publicly available. When we did our initial vaccine screen, we had used transcript data from human CSF from 2 patients to pick antigens, and an additional publication since then has expanded that data to include CSF samples from 33 different crypto-positive patients, and those are what's graphed here. Looking at the data from human samples is important because we do want to understand how the bug acts in a human host, but the data that's available for humans is from after the infection has already disseminated. So I also looked at another data set for samples from mouse lung 7 days post-infection. And the mouse lung data is important because you can actually analyze lung samples during infection, which is where you're going to want the vaccine to work. An effective vaccine won't actually allow for disseminated infection. So both the human samples and the mouse samples, this is all RNA-seq data. So we're assuming that transcript abundance is a correlate of protein abundance here. So I took these interesting data sets that were available on GEO, and I put together a graph to try and better understand the idea of cross-reactivity during vaccination. So the y-axis shows the rank of the transcript. So something that's ranked as 1 is the most abundantly transcribed in that sample, where something with very few transcripts could be ranked as like 6,000, for example. And Cda1 and Cda2 both give us 100% protection as vaccines against a wild type challenge. And those genes are highly expressed in both human CSF and in the mouse lungs. So you can imagine if Cda1 was not expressed for whatever reason, that there might still be plenty of Cda2 around to make a decent vaccine target. And you can see that Cda3, which is not nearly as protective as Cda1 or Cda2, it's still in the top 50% of transcripts. It's just much lower than Cda1 or Cda2. And then we have FPD1, which is not protective at all as a vaccine protein, but it's transcribed far less than the other family members. So this just gives a little bit of a broader context to the idea of cross-protection. So we wanted to see if we could extend our work to another family of proteins, and we chose to look into the carboxypeptidase family. CPD1 is a capsule-associated protein, and it's abundantly transcribed during human infection. And it is extremely homologous to 2 other proteins that we're calling CPD2 and CPD3. CPD1 is a protective vaccine antigen in our model, but we have not yet tested the other 2 as vaccines. So we picked this to look at because the top 2 homologs share a lot of identity with CPD1 over the entirety of the protein. These are much more similar to each other than the proteins in the CDA family are, and they're very similar in sequence, and I thought this would give us a pretty good shot at seeing cross-protection. So we went back to our survival model to look for cross-protection within this family. And the unvaccinated deletion strain and the unvaccinated wild type strain die within the same time frame. When we vaccinate mice with CPD1, we get pretty good protection against the wild type challenge. If we vaccinate with CPD1 though and challenge with the CPD1 Delta strain, we completely lose the protection that the vaccine gave us against KN99. And I found this to be kind of surprising. Like they're so similar in sequence that I thought CPD2 and CPD3 expression would be enough to induce a protective vaccine response. But even though they are so similar, like I mentioned, we haven't yet tested the other 2 antigens as vaccines. So we don't even know if those 2 are protective on their own. And our knowledge of the antigens in the CPD family is less than what we know about the CDA family. So I went back to the transcriptomic data to try and make sense of the survival results. And for CPD1, which we know is a protective vaccine, there is greater expression in the mouse lung than there is in the human CSF. And CPD1 is really at the top of the list for transcripts in the mouse lung. There's not much of a difference between expression of CPD1 and CPD2 in human CSF, but there's a big drop in CPD2 expression compared to CPD1 in the mouse lung. And that holds true for CPD3. There's over 10 times more CPD1 transcripts in the mouse lung than CPD2 or CPD3. So there could be a cross-reactive vaccine response, but CPD2 and CPD3 just might not be expressed at high enough levels to make them protective target antigens. So we'll definitely be studying this family in the future to try and get a better handle on this. I'm just going to give you a brief recap of what I have shown you. So our survival studies have shown us that cross-reactivity is necessary, but it's not always sufficient to induce cross-protection. So we do see cross-protection within the CDA family, and the protective CDAs are highly expressed in the mouse lung, and we do get a cross-reactive IgG and interferon gamma response within this family. We do not see cross-protection following vaccination with CPD1, but there is lower expression of CPD2 and CPD3 in the mouse lung than there is of the really abundantly transcribed CPD1. And we do have experiments ongoing to look at cross-reactivity between the CPD family antigens. And then just kind of a big picture concept, it raises a question of, is targeting protein families something to keep in mind when selecting vaccine antigens? Because if you target a protein family, then you theoretically have more immune targets, and then you have a better chance of retaining protection if mutations develop. And I'll wrap things up here with the cross-protection model we've come up with. There's all sorts of ways you could choose to group cryptococcal antigens. But for these purposes, we'll consider them to either have homologs or to have no homologs within crypto or to belong to a protein family. So after vaccination with an antigen that has no homologs, like MEP1 that I showed earlier, we can challenge with wild type cryptococcus. And this induces a response specific to that antigen. And this can include CD4 T cell activation, interferon gamma production, and antibody binding. And all of these things lead to eventual fungal clearance. If we vaccinate with that same antigen and then challenge with a strain with the corresponding deletion strain, that antigen is not present for the immune system to recognize, and we do not trigger those protective responses. And this makes for an ineffective vaccine. If we look at our other group of antigens, which are antigens that belong to protein families, we can vaccinate with just one of the proteins in a family and give the mice a wild type challenge. The vaccination with a single antigen can lead to reactivity to that antigen, but also to other antigens within that family. And we'll eventually get fungal clearance. Using the same single antigen vaccine though, we can also challenge with deletion strains, single deletion strains, where the other family proteins are still expressed. So we don't get a response to that specific vaccine antigen, but we can still get a response to other family members. And this can improve survival in vaccinated mice over unvaccinated mice, like I showed with the CDA family. But if the expression of the other family members is not sufficient, like with CPD1, then we actually won't see cross protection. And this model is still in the works. And I just want to thank everyone who I work with who has helped me with this project. I obviously want to thank the organizers of the series for giving me the opportunity to share my work, especially Jess Brown for reaching out to me. And I showed lots of science, so I obviously have to balance it out with some cute dogs who are stuck listening to way too many of my practice talks. And my contact info is on the bottom right there if anybody wants to talk about anything else.
Thanks, Maureen. That's really fantastic work. And so there are a couple of questions in the chat. We'll get started with some questions for Dror, but please continue entering things into the chat. Dror, I get the honor to ask the first question. And so I'm very curious about how your filamentation promoting TFs block the signal from the RIPs. So you just sort of mentioned in passing that there doesn't appear to be a displacement. And so I was wondering if you could expand on that, whether you think this is post-transcriptional or what's going on there.
So it's a great question. We haven't tested the RIP binding directly. I haven't done any chromatin immunoprecipitation. I do know that at least some of the RIPs are still accumulating fine, even though the cultures are going through filamentation. It could actually be that the presence of other transcription factors could maybe displace some of the RIPs. For instance, I mentioned PAC2 is also a Whopper transcription factor. There's a kind of a theory that maybe it could displace in at least some key genes the binding of RIP1 to those genes, to those promoters. That's one theory. We haven't really tested it. We do also know that there is an enrichment of RIP targets in the genes that are downregulated in response to cyclic AMP. So it could in fact be that, so that there is a decrease in the genes that are normally bound by the RIPs. So it could be a matter of binding. It could also be that some of those genes are, that some of those transcription factors are able to activate, the expression of genes that normally would not be there, and that's sufficient as well. I hope that answers the question.
Yeah, that's very cool. Thank you. All right, Liz, do you want to take the first question from Maureen?
Yeah, Maureen, thank you so much for that really interesting talk. So the first question comes from Mark Swigerdal, who asks, he says, beautiful work. He was wondering wondering if you determined also the fungal burdens in the different organs to see if vaccination with your candidate will induce fungal clearance and if this is correlated with survival.
Yeah, so with vaccination and then challenge with a wild type strain, in the mice that survive, we don't see dissemination to the to other organs. So if the vaccine is protective, we don't see dissemination. I haven't looked at time points other than end of study for the vaccine, like CDA1 challenge with CDA1 deletion strain, but there is no dissemination at the end of the study. The mice that do not survive, I have not looked at the fungal burden in other organs. And then in terms of deletion strains, if like knockout mouse strains.
Let me just ask that question so everyone can hear what it is before
Okay,
you answer. Yeah, sorry. Yeah, I know you could probably read it and prepare your answers ahead of time. So Mark goes on to ask, will there be protection after vaccination in B cell or T cell knockout mice, such as MU-MT, CD4 KOs or RAG1 KOs?
So yeah, we have done survival studies with, again, vaccination with wild type challenge, not with vaccination with deletion strain challenges. And in our model, if T cells are depleted before challenge, but not during vaccination, other people in the lab have done this where you still see protection. But if we use like MHC2 knockouts, where there's no functional CD4 T cells, then we do not get protection at all. So we need to have CD4 T cells during the vaccination phase, but not necessarily during the challenge phase. And then MU-MT knockouts, it's actually kind of specific to each vaccine. Some of them survive like wild type, and then other antigens, it depends on the vaccine whether or not there is a difference with the B cell knockouts.
Interesting. There's a bunch more questions for Maureen in the chat, and I've also got one. If anybody has questions for Dror, sorry, I'm mispronouncing your name, please go ahead and put them in there. I'll just keep going with Maureen until those come in. Jenny Lodge asks, she says, nice work and great presentation. 2 questions. CDA1, 2, and 3 are all GPI anchored and are in the cell wall, but FPD1 doesn't have a GPI anchor, so it may be secreted. Do you think this could be a contributing factor in what you saw?
Yeah, I'm not sure specifically for the CDA family because we do have successful vaccine candidates that are for secreted proteins that don't have GPI anchors. But yeah, that's a good question. I would have to think about that.
Okay, she goes on to say, Fred Dietrich at Duke is looking at structural similarities rather than amino acid homology. So rather than the primary structure, the folding of the protein. And she's wondering if this would be a good methodology to identify homologs that would be structurally similar, but not sequence similar.
Yeah, I think that would be something to look at because we have looked some at peptides. So we have some idea of like what are the protective regions, and we can see cross-reactivity with peptide vaccination. But yeah, rather than just sequence, I think that actual structure would be really important. I just don't know a lot about it.
Yeah. I had a question for Dror, so I'm just going to ask it. It's my prerogative. So you kind of suggested, you had this model at the beginning of your talk, which is that room temperature, or room temperature, right, triggers this process, and that might be an environmental signal. And you had a sort of a positive activation framework for your pathway. Why is it that you thought room temperature is the signal rather than lack of temperature being lack of activation? Does that make sense? So can you turn it on its head a bit? Is there data suggested that it's one versus the other?
It's an interesting question. So you're asking if, in fact, it might be that 37 is repressing this instead of room temperature.
Exactly.
I don't think I have I need to think about this more because I don't think I have an immediate quick answer. I don't think I have a good way of differentiating between lack of 37 degrees and or room temperature. We do know, like, there is research in our lab that is looking into by a very talented grad student, Anna Morrison, who is looking at kind of the threshold at what point you get filamentation, and even if you go down only a few degrees below 37 degrees, you do have some at least initiation of filamentation, which might imply that 37 is like you might be suggesting, it's kind of more blocking a certain pathway rather than room temperature inducing it. But I don't think we have a great answer to that question.
Okay, thanks. Jessica, do you want to ask the next one?
Yeah, so the next question is sort of builds on that. And so my question is, how much variation do you see in these transcription factors across histostrains? And then sort of related to Liz's question is, do the strains from tropical regions have different temperature barriers, or is 37 sufficient to keep things in the yeast phase for basically all strains? So strains that might have a more endogenous temperature that's higher, do you still see the same cutoffs, or do you see different cutoffs? And does that then correlate with variation in some of these controlling transcription factors?
So the strain that we're working on is that all of my work has been on is called North America 2. So I supposedly also has, kind of, it has more variations in temperature than a lot of the other strains, but I'm pretty sure, if I remember the literature, that all of the different strains of Histo have a similar transition. Room temperature and 37, I'm kind of maybe shooting myself in the foot here in a way, but it's a signal, but it's not the only signal that promotes fermentation or yeast growth. So there obviously needs to be a set of stimuli that kind of push to one direction or the other. But the temperature has just been one that's been easy and sufficient to push for one morphology or the other. Again, I haven't looked at the other strains, and that's an interesting point. I need to look at how it would be interesting to look at how conserved those transcription factors. I mean, they're there for sure, but how variable they are in other strains. I don't have a great answer to that. No.
Yeah, I think I was at a talk last week for the CanFundNet and got to see a really nice talk about sort of desert fungal species. And a lot of these guys are surviving in like 57 degrees, right? So like quite high compared to what we would consider to be a high temperature. I think there's this idea in medical mycology that human body temperature is a threshold past which most fungi can't survive, right? And that's just not true, unfortunately. But that maybe this is that 37 is actually below that. It's cold, right? For some of these organisms. Maybe there's an intellectual bias that we have there. Maureen, I've got another question for you, which is the idea of vaccinating against protein families seems like a really promising one. This is from Jessica. What is the sweet spot in terms of conservation? Do you see a lot of strain variation in any of these proteins? And could some of these new mRNA vaccine technologies be used to account for genetic drift in antigen proteins by covering those possible variants?
Yeah, so I'm not sure about the variation between strains, but so all of our CDA1, CDA2, and CDA3 that I showed were based off neoformans. And we have actually the same constructs for Gadii R265. And what we see is we actually can't really protect against R265. So if we vaccinate with CDA1 or CDA2, CDA3, we can extend survival a little bit, but the mice do succumb to the infection. And that's with protection of Gadii with the Gadii recombinant proteins. We can vaccinate mice with Gadii recombinant proteins and challenge with neoformans, and we still get protection just like it was with the neoformans vaccine. So Gadii proteins can protect against neoformans, and we can still see like this very similar antibody response. So I'm not positive on like strain to strain, but across Gadii neoformans species, we do see cross protection, but going only in one direction, even though there is some variability between the sequences. And sorry, there was something about mRNA that I already forgot.
I think the question is whether, so if you had this idea that there would be mutations happening, and that giving working with a protein family would help allow cross protection, right? But maybe mRNA vaccines can help sort of combat that as well. Does that explain the question better?
Yes,
Yeah.
just since you can put so many different variants in a single vaccine more easily than you can with protein. Can you expand on your protein family idea and just cover genetic variation as well?
Yeah, that's it. I haven't thought much about the mRNA vaccines in terms of a fungal vaccine. I was thinking about mutations more like because we haven't seen a fungal vaccine before. So if we are vaccinating, is this going to put pressure on the bug to mutate? And I don't know if that will be as much of a problem as I actually think it is, but it would be really cool to start to look at the mRNA platform, especially since that is like the hot new thing right now.
Yeah. All right, if anyone has any questions, feel free to turn on your mic. Otherwise, thanks to both our speakers for really great talks. I enjoyed them a lot.